Hello and welcome back everyone to more biotechnology. So now that we have some good fundamental principles under our belt, we can devote this chapter to revisiting some concepts that we talked about in chapter 1 now that we've added some tools to our tool belt. So this first video is going to do a lot of that revisiting, and then later sections will add further to our toolkit by describing the use of assays and different techniques that will help us on down the line. So let's go ahead and get into this. So we've said this several times before, but most biotech products are going to be proteins. But even if they're not proteins, biotech products in general come from cells. They come from living cells. So if you're going to develop, manufacture, and market a biotech product, you're doing that with the knowledge that you need living cells in order to produce that product for you. So one example that we've looked at in the past is the case of insulin. So if we're going to sell insulin on the market, we've got two different choices at our disposal. Now, we could take the hard route and we could harvest the pancreases of an animal like the cow that is going to be butchered for its meat anyway, and we can grind up the pancreases and purify the insulin contained within, but why would we want to do that when we've already established that we can use recombinant DNA technology to transform bacteria with the insulin gene and get them to crank out a whole bunch of this insulin much more efficiently and at a much lower cost? The other example that we've looked at in the past is TPA, which is a signaling protein that is produced in humans, but it's produced in very, very, very small quantities. So naturally, by, again, using recombinant DNA technology, we can make it in much, much, much greater quantities. So these are the two examples that we've looked at in the past, but the same principle should apply to just about any sort of biotechnology venture that you want to get into. Biotechnology is all about taking processes that are already occurring in nature, processes that you want to take advantage of, and use what you know about how cells operate, how cells make proteins and other molecules, and trick cells into doing those things much more quickly and much more efficiently than they occur in nature. Now, a new example that your book is putting forth in this chapter is the case of an enzyme called amylase. Amylase as an enzyme is going to be a protein. So we've said in the past that almost every enzyme is a protein itself, with a couple of exceptions. So amylase is a protein, and as such, the production of amylase by cells is going to be governed by the sorts of things that we talked about in chapters 4 and 5, both transcription of DNA and translation into a fully fledged protein. Now, what amylase does, the enzymatic reaction that it catalyzes, is it breaks down plant starches into the disaccharide maltose. So if you're not quite if it's not quite clicking what a disaccharide or what carbohydrates are, you may want to go back to chapter 2 in which we discuss those things. So once we have our disaccharide maltose, which is just two glucose monosaccharides joined together, another enzyme called maltase will then break that down into two molecules of glucose. And glucose is going to be heavily utilized by cells throughout the body for making energy. So amylase represents a very high potential biotechnology product, something that if we wanted to, we could develop technology to mass produce it, and we could sell it and market it. So if we were operating a biotech company and we were interested in doing this with amylase, what sort of factors should we consider? Because we've said in the past, you need to consider things like how easy is it going to be to make this product? Is it really in our wheelhouse or in our expertise? How much is it going to cost? Is it going to be safe? And things of that nature. So the first thing for us to consider is the market size. Basically, how much are people really going to need or want this product? So if we're going to invest our time and our capital in amylase production, we need to be convinced that there is going to be sufficient and widespread demand for it. The next thing for us to consider is the sources and ease or cost of production. So basically, how easy or difficult will it be for us to make this, and will we be able to turn a tidy profit on it? So it's one thing to have a good sizable market for our amylase, but that may not end up mattering if it's too difficult or costly to produce it. So that is yet another thing for us to consider. So in this case of amylase, in reality, there is a very good widespread market for the enzyme. So there are many different applications for the use of amylase. There are many different ways in which you can take advantage of an enzyme that breaks down starches. So you can look at the textbook for a few of those examples. But as we've said before, if you're producing a protein, then probably one of the best ways to go about mass producing it is to transform bacteria or other cells with the amylase gene and then get them to mass produce it recombinantly, the same way that we saw with insulin and TPA. Now, a lot of what biotech companies do, and keep in mind this is just the kind of the bare bones analysis of it, when they're trying to determine a product's potential and whether they want to go and pursue that product, they go through what's called a comprehensive product development plan. So this is the same sort of thing that we've talked about in the past, but some of the questions that we may want to ask as we go through our CPDP is, number one, will this product meet a critical demand? So that leads into the market size thing that we talked about on the previous slide. Again, how large will our market or customer base be? Number three, which is something that we have not discussed in this particular video just yet, does our in-house data suggest the product will work? So no biotech company is going to launch into investing in a biotechnology product if they haven't already done some preliminary research and development on determining how effective or how good and safe it's going to be. So in addition to needing to have preliminary data that says, okay, this product is going to do what we say it's going to do, you also need to be convinced about things like it's not going to make people sick, it's not going to have unusual side effects, like in this case causing someone to grow a third eye, which is kind of extreme. So we need to not only be convinced that our product will do what we say it does, we also need to be convinced that it's not going to do undue harm to people who buy it. Number four, something that we need to consider is, can we secure a patent for this product? So this one is particularly important because this really is going to determine how much of the market share that we get for our particular biotech company. So any company that's selling a product doesn't want to share the market with a whole bunch of other companies that are making the exact same product. So if we can patent our product and maintain that patent for as long as we possibly can, we reserve the right to be the sole distributor of that product. And if anyone else wants to sell and distribute that product, they're going to have to pay royalties to us. However, if we either can't secure a patent or we let a patent that we currently have expire, then that particular product becomes fair game to become generic on the market, meaning any company can make that product and sell it and maybe undercut our current price for it so that it cuts into our market share and makes it harder for us to sell it and market it. And then finally, something that we have discussed, can we make a profit on this product? There's no point for a company, even if making and selling the product is a noble effort, even if it's something that the population drastically needs, it's just not going to be tenable to continue to sell and make that product if we're not making a profit on it, because if you're not doing that, that's a good way for a company to eventually go under. Alright, so that's going to do it for this first preliminary video in which we revisited some concepts regarding how biotech companies operate, things that we've talked about in the earlier chapters in this course. So I thank you for your attention, and I hope you'll join us next time, and we will start talking about assays, which more or less are just techniques or tests that are done in the laboratory to determine things like concentration of a particular molecule, potency of a drug, and things of that nature. So bye-bye for now, and I will see you next time.